Hello everyone, I am Annie. Today I'm going to talk about the mitosis process I learned in Bio 110 class at Kunshan University. So our lecture today will mainly be separated into three parts. The first is about a lecture on mitosis, and then we're going to have a lab experiment, and last I'll be giving some questions about mitosis. First lecture on mitosis. Before we delve into the mitosis process, I want to talk about the significance of mitosis and the difference between two similar concepts, mitosis and meiosis. Mitosis is essential for growth, development, and tissue repair in multicellular organisms. It generates two cells identical to the parent cell, maintaining genetic continuity. And for mitosis and meiosis, they're totally different. Mitosis produces two genetically identical diploid daughter cells occurring in somatic cell for growth and repair. However, meiosis produces four genetically unique haploid gametes for sexual reproduction. And then we'll have a look in the cell cycle. In the cell cycle, actually 90% are made up of the interphase, which can be further divided into three phases: G1 phase, or the first gap, an S phase, senescence, and the G2 phase, which is also called the second gap. And the cell grows during all three phases, but chromosomes are duplicated only during the S phase. And the rest 10% of the cell cycle is actually the mitotic or M phase. Okay, then we're going to talk about the specific stages of mitosis. First, it's about the interphase. Also in the G2 phase, the centrosomes replicates during G2 interphase, forming two centrosomes that migrate to opposite end of the cell during prophase and prometaphase. As you can see in the figure 3, there are already two centrosomes. And in prophase, the mitotic spindle is a structure made of microtubules that control chromosomes' movements during mitosis. Anaster extend from each centrosome. Anaster is a radical array of short microtubules. Then we're going to move on to the prometaphase. In prometaphase, some spindle microtubules attach to the kinetochore of chromosomes and begin to move the chromosomes. Kinetochore is a protein complex assembled on the centromere. Okay, next we're going to move on to the metaphase. In metaphase, as you can see in figure 6, chromosomes are all lined up at the metaphase plate, a plane midway between the spindle's two poles. After metaphase, we are going to enter the anaphase. The cohesion are cleaved. The cohesion is the kind of stickiness between two sister chromatides. Okay, the cohesions are cleaved and sister chromatides separate and move to each centrosome by the microtubules shorten. At the end of the anaphase, duplicated groups of chromosomes have arrived at the opposite ends of the elongated cell, and the cell is ready to separate it into two. The last step is the telophase and the cytokinesis. Cytokinesis begins during anaphase or telophase, and the spindle eventually disassemble. And new nuclear envelope and nucleus start to form. So in this picture, we can see all stages of mitosis. So you may want to pause here and see if you can connect all these stages. Next, we're going to move on to an interesting part, which is about a lab on mitosis. So today's lab is about observation of mitosis in cells. We're going to use the garlic roots in our experiment. First, cut a few roots and place them into a glass container filled with fixative solution. So when we cut it 1 centimeter, that is because we have to observe the mitotic region in the root cell, which is in the growth zone. The growth zone is about 1 centimeter from the start of the root cap, so that's why we make it 1 centimeter. And why we use the fixative solution is because the fixative solution rapidly penetrate the cell to preserve their structure, which will make it easier for our further observation. And next, move 4 to 5 root tips to a glass watch and add 4 to 5 drops of 1 hydrochloric acid in the glass watch to cover the root tips and allow the root tips to soak in the acid for 5 minutes. So the hydrochloric acid is actually a dissociation solution which can break down the pectin between cells, separating them for easier staining and observation. And after this step, we're going to use water to gently wash off the acid for 2 to 3 times. Okay, next step, we're going to add 2 to 3 drops of 0.8% genetium violen stain to cover the root tips and allow the root tips to soak in the stain for 3 minutes. Then use water bottle to wash off the excess stain 2 to 3 times. Staining makes the cell nuclei and chromosomes more visible and coloring them in a purple color. Last, we're going to place 1 root tip on a clean glass slide, add 1 drop of distilled water to the root tip, use eraser end of a pencil to apply pressure on the cover slide to squash the root tissue. This step is very essential because it actually depends on whether you can observe a single layer of cell, which is very important on whether you can see the cell clearly, because if you don't press it hard enough with a eraser at the end of the pencil, you may see that the cells are all in double or even more than 3 or 5 layers, which will be difficult for your observation process. Okay, so this is what we expect to see in the experiment. Let's observe what stages can we see. So first, this is a cell in the interphase. The cell in the prophase. You can see that the chromosomes condense and the spindle begin to form. Next, we can observe also a cell in metaphase. Chromosomes align at the metaphase plate. And then it will be the anaphase cell. So in anaphase, sister chromatids separate and move to the opposite pole. Finally, we move on to the telophase, and it involves the reformation of nuclear envelopes and cytokinesis. Okay, we'll move on to the next part, which is about the questions and answers on mitosis. So first question, how does prophase contribute to mitosis? So in prophase, chromatin condense into visible chromosomes held together by centrosomes. The nuclear envelope breaks down and the spindle starts to form. Centrosomes move to the opposite pole, setting up the framework for chromosomes segregation. This stage prepares the cell for the subsequent steps of mitosis. And another question, what is the significance of metaphase in mitosis? So metaphase is also crucial for ensuring the proper chromosome alignment. Chromosomes line up at metaphase plate, and each sister chromatid is attached to the spindle fiber from opposite pole. This alignment ensures that when separation occurs in anaphase, each daughter cell will receive an identical set of chromosomes, maintaining genetic stability. Then we'll move on to some advanced questions. So how do cancer cells bypass the normal regulatory mechanism of mitosis? Well, you can pause the video here and think about the question yourself, and then you can see if my thoughts are similar with yours. Normal cells have checkpoints that ensure DNA integrity and proper mitotic progression. However, cancer cells often have mutations in tumor suppressor gene like TP53 or RB1, leading to uncontrolled division despite DNA damage or incomplete replication. This answer is based on some researchers in 2019. Okay, so that will be the end of today's lecture. Thank you so much for listening. And here are the references, all the sources that I used. I'll move myself here so you can see all of the references. Okay, thank you again. Goodbye.